Hey everybody, how's it going? We're gonna do our last set of evolution notes here, which are the classification ones. Also, we'll learn how to make cladograms. Okay, so let me swap that. So, big ideas: the pattern of life's diversity that has developed during life's history on Earth. How do we determine evolutionary relationships between organisms, and what are the characteristics of life that are useful for determining evolutionary relationships? So, few things to dig into here. So, old school way, what we used to call taxonomy, um, was kind of developed by John Ray. He was kind of the pioneer, but Carolus Linnaeus, if you remember him from the other set of notes, he gets a lot of the credit. He came up with that naming system, that binomial nomenclature you see here on number two. It's basically just grouping species by similar, characteristics, mainly physical characteristics, because that's all they could, really do back then. So he came up with the naming system, like the genus species system that we still use today. And you can see down here in the left-hand corner, Homo sapiens, which is what we are as humans. So a little more about old school. Um, Now it's what we call, we have the overall heading of all of this is called domains, and that's actually a little bit new school because it was not around when I went through high school and college. And I'm not that old, but not that old. Um, if you look at the bottom, there were five traditional kingdoms. It was always Monera when I went through school, and that was all the bacteria kind of lumped together. And then the other four are still the same, the protists, the fungi, the plants, and the animals. Um, and then at some point they added three domains up above the kingdom. So you've probably heard kingdom, phylum, class, family, order, genus, species. So you've probably heard that list before. Well, now domain is up at the top. Okay. And at one point, these kingdoms were based on like nutritional modes, and that also is not really valid anymore. Things have changed. So here um, you can kind of see all of the list over here, the taxonomic group, domain, kingdom, phylum, class, order, family, genus, species, all the way down. And we're looking at the gray wolf. So it's in domain Eukarya, along with 4 to 10 million other species. Okay, and here's the wolf down here. Animalia. So now we kicked out anything that wouldn't fall into that category. And then the chordates. We've kicked out everything that doesn't have like a nerve cord. And then the mammals. Now we kicked out everything that's not a mammal. Okay, and then carnivores, and then you get down to like the family, genus, species. We're getting more and more specific as we go down this list, right? Until we get down to just the genus species of the gray wolf. Okay, so that's kind of how classification works. Probably have seen that before. Here's just a little diagram of the domain. So you have three domains. Bacteria, which sometimes you'll see an EU in front of that, called eubacteria, and then archaebacteria. So the first two domains are both bacteria. Eubacteria are kind of the everyday bacteria. They're found in, they're found on your tables and on your skin, and just kind of everyday all around. Even within the bacteria within us, most of it would be eubacteria. Archaebacteria live in really extreme environments. It could be like super salty, it could be super acidic. All these environments that other things can't live, archaebacteria can live. And then the eukarya, right? These sound like eukaryotes. If you remember anything from cell biology, there's prokaryotes and eukaryotes. The prokaryotes are your bacteria over there, and then your eukaryotes are the other ones. And everything else falls under eukarya. Your protists, your fungi, your plants, and your animals are all in the eukarya domain. Not a table you have to memorize or anything. I just thought it was interesting. It's actually from an older textbook, but it is kind of interesting to look and compare. Um, between some of these things and how things, deal with how the three domains have their chromosomes set up. So bacteria are both usually circular, eukarya is linear. You have your nucleosome structure, chromosomes, your introns. A lot of this is genetics that you guys haven't probably heard yet, but you're gonna. Later this year, we're going to talk about operons. We're going to talk about capping of the mRNA. RNA polymerases are going to become old news. Like, we're going to do all this stuff. But it's kind of interesting just to see what some of these differences are between them and how they get separated. Again, just a picture about, a bunch of bacteria. You see the deer in the middle of the picture there. and all the arrows coming off showing you like all these different sorts of bacteria that are either on the deer or in the pond. And those are all eubacteria up top. And at the bottom, you can see down there in the hot springs, looks like they're the ones at Yellowstone, the Mammoth Hot Springs. You have an archaebacteria down there that lives a really extreme environment. Another table comparing the kingdoms. So the protists, the fungi, the plants, the animals. Again, how do we separate these guys? Well, it's based on, some pretty typical characteristics. Unicellular or multicellular. Does it have a cell wall? If so, what's it made of? You guys should know that from our biochem chapter. What sort of cell and tissue types do we have? Can it photosynthesize? What's the organic molecule situation? So just, again, nothing I'm going to make you memorize, but just kind of an interesting thing to check out and how they separate the kingdoms. New school, so we're picking up a little bit here. What was wrong with the old school? Well, things kept changing. So the number of kingdoms have changed over time. At first, there were just two. It was just plant and animal because they didn't have microscopes. They didn't know about all the small stuff. And then it went to three, and then it was five, and now it's six. Like there's just been so many changes. So early work was based on observation, which looks can be super deceiving, as you can tell down here with our marsupial mole and our placental mole. They look pretty much both like moles, but one being a marsupial and one being a mammal, they actually operate a little bit differently. So most species that get described are macroscopic. They're big. A lot of species are actually microscopic, right? They're actually really tiny. So it's easier to describe the big ones. It's easier to see the structures. It's easier to find them. So macroscopic is described a lot more than microscopic. Enter DNA. Now we're gonna, start getting into the new school stuff. We have DNA, and you guys have been poking around the blast lab and seeing how related sequences are, and that's kind of how things are done now. So remember DNA, if they're very closely related, they're going to have similar DNA. As species diverge, as they get more and more away from a group that they used to live with, their sequences will also diverge. They'll be put under different evolutionary pressures, and they will change differently, and they'll have different mutations and things like that. So the more the sequence is different, then the more they have split apart. So and this diagram down here is not too big a deal. It's just kind of, I'll get into some details about diagrams like this coming up. But it's just looking at the relationships, the evolutionary relationships among organisms, which we call phylogeny. So this is just kind of a family tree of different birds down here. So because we can do this, because we can track DNA now and we can look at mutation rates, we can kind of use them as a molecular clock. And so different genes and different organisms have different mutation rates. So depending on how fast things change and how similar or different they are to other things, we can kind of date things back and see how things are going. On the left over here, based on the length of line, you can see, how much time it was, how much time has gone by since mutations, and which things are more and more similar and which things aren't by the length of line. And on the right, you can see, we have time across the bottom here in millions of years. And then we have nucleotide substitutions on the left-hand side. So we're measuring nucleotide substitutions, which is just another term for mutation, right? When nucleotides, ACGs and Ts, get substituted, that's a mutation, okay? So if you look, the line's very linear, right? So, over time, mutations have been pretty constant as far as how often they occur over time. And so things keep changing. That's some more, evidence for evolution and things changing over time. Nothing too big here. Don't get bogged down in all the different types of bacteria at the top of that diagram or anything. It's just kind of interesting that DNA has identified these endosymbiotic ancestors. This is how we knew about mitochondria and chloroplast. And if you look back at this, some of the timeline down here of what's gone on, here's basically where the mitochondria was and when it was brought into the cell. And then you see the cyanobacteria, a little bit about red algae and green algae. And these are based on DNA. They could tell how long ago, based on the mutations and how similar the sequences are, how long ago they would have diverged from some of the other species that are still around, so they can kind of put a clock to it and see where things diverge. So it's pretty cool. Cladograms, cladistics is the study. Cladograms are kind of these diagrams you guys have already been dealing with in the BLAST lab. So you're probably a little familiar with looking at them already. So what you do when you make a cladogram is you look at characteristics that things share, and you can still do that using DNA. So we have a couple of different ones. We have what's called a shared derived characteristic, a trait shared by a group of organisms, but not with a distant ancestor. So for instance, mammals have hair, all of the mammals, so that's the group, all of the mammals have that trait that they share, that they have hair, but they don't share that with other common ancestors. So, birds don't have hair, and other organisms, reptiles don't have hair, even though they're all animals. So it's kind of when something breaks off and it's another characteristic that that group may share that the common ancestors do not. On the shared primitive characteristics, this is the trait that's shared with a distant ancestor. So things like backbones. So mammals and reptiles and birds and other organisms have backbones, as does their common ancestor. So those are traits that you can trace back far longer than you can the shared derived ones. If it derived, that means it came later. Okay. Simple cladogram here. If you look at the bottom here, so you have a notochord right here, okay? And so you have just this guy over here with the notochord, and that's all he has because he has the red line. And then you have the vertebrae here in the red and blue. And so you have this guy with the red and the blue, and I believe that's a lamprey. And then you have the hinged jaw, so we got another kind of fish up here, and he's got all three of these things. And then you have the tetrapod, tetrapod feet. And so you have some sort of reptile or lizard here, and then you have mammary glands being added. So only the little bunny right here has the mammary glands, but it also has all of this other stuff. So that kind of shows you one way to do a cladogram. I'm going to show you another way, though. So how to make one? So the first thing you have to do is figure out how many species have a characteristic in common. So what you would check out with this, so over here you have your characters, right? You have your hair, your amnion, which is like the sac in an egg. You have 4 walking legs, you have hinged jaws, and then you have the vertebral column. And then you have your taxa, or your organisms across the top. So we have leopard, turtle, frog, bass, lamprey, and lancelet, okay? So in this case, the lancelet doesn't have any of them, okay? It doesn't have any of them, and it's called the outgroup, which I'll get to in a second on the next slide or two. Lamprey just has the 1, bass has the 2, frog, 3 of them, turtle, 4 of them, leopard, all 5 of these guys, okay? So you can kind of see like how things may have evolved and which ones would have been more related to others just by looking at what characteristics they have in common. You just put a 1 if they have it and a 0 if they don't. So this is what then the cladogram might look like. If you take the data on this slide and you put it over here, the lancelet being the outgroup, and you can see at the bottom, species most closely related to the in-group. And the in-group is just the group you're studying. So in-groups and out-groups, they're kind of a little bit, not necessarily arbitrary, they're not arbitrary to the person doing the research, but whoever's doing the research gets to kind of pick the group they're looking at, and that becomes the in-group, and then whatever's next to it would become the out-group. Okay, so you can see based on how the drawing goes here, the vertebral column, the hinged jaws, right? So anything beyond the hinged jaws, right? Anything that comes up here, so the bass and the frog and the turtle and the leopard would all have the hinged jaws because it evolved here. So that means everything from here forward, okay, is going to have the hinged jaw. Okay, and anything before this, so the lamprey and the lancelot would not. Okay, so that's just kind of how you read a cladogram. More on how to make it. So we apply what's called parsimony or Occam's razor, if you've ever heard of that, which basically means all things being equal, which can be an assumption because things are not always equal, but all things being equal, the simplest explanation is usually correct. That's Occam's razor. Okay, so characteristics are most likely to evolve once. Okay, that would be the simple thing, right? Saying, hair only evolved once, or whatever characteristic you're talking about only evolved once. And so if you look at this data down here, the percentage differences between sequences. So the human is, obviously no different with another human. So we have a human here and a human here, and there is no difference. A human and a mushroom, we have about 30% difference in our sequence from a mushroom and about 40% different than a tulip. Okay, and you can see the mushroom and tulip together are about 40% as well. So if we go to the next slide, now we kind of start putting them, what would the cladogram look like? Well, we apply this other rule called the rule of maximum likelihood. Assuming mutation rates are similar or the same between the human and the mushroom and the flower, which again is a huge assumption because they're not always the same. Some things mutate quicker than others, but assuming they are the same, more closely related organisms will have fewer differences in the genetic sequence, right? If they evolved, if they are more similar organisms, their DNA would be more similar. If their DNA was more different, if the ACGs and Ts, if we had a bigger difference in that, then they probably evolved earlier and have now split. They've now diverged over a longer period of time. Okay, so you can see down here, tree 1 is definitely more likely than tree 2. You can see the human and the mushroom are both around 15%, which would be much more similar to each other than over here with the person and the tulip. They're not quite as similar as far as the sequence difference would go. So 1 is more likely, 2 is less likely. Other cladogram basics. All species are placed on the tips of the tree, not the branches, so we usually write them at the very ends. Each cladogram branch point should list the shared derived characteristics, so that is right here. So at each branch, you're going to, put down what characteristic is the new separator, okay? And then all shared derived characteristics appear together only once in the cladogram, unless you know they arose independently. We did have one of those in our slides earlier in the chapter where we said that wings arose independently, right? So you might have, wings at this branch, and you might also have wings at this branch, later on if you were dealing with wings. So it can happen that way, but typically things only arrive one time. Let me back up real quick. And generally, the cladograms that you've seen in your lab, in the blast lab, they're easier to draw, and I'm totally cool with that if you want to draw them like that, which is really just a diagonal line like this, and then you have the line coming off and the branches coming off like this, okay? And so you would have your shared derived characteristic down here in between them, and then you would put your organisms up here at the top, whatever those might be. And you might even have a branch in here because there was something else that happened here, some other trait, and these two are really closely related. But you're totally welcome, and I even suggest that you draw them like that, okay? So here's how to make one again. You have some data here, so try this one. Which tree do you think is best? You have species 1, species 2, and species 3, okay? All of these birds, they all have things kind of in common with each other. But do you think tree 1, okay, is this one or this one or this one most likely to be the way it occurs? Well, it's pretty tough to tell. You can pause the video and look at the birds for a minute if you want. It's pretty tough to tell. What else would you want to know to figure this out? If it were me, I would love to have some DNA. I'd love to have some sequence to go by, right? So now we have some sequence and we have some things to compare. Okay, so here's species 1, okay, and here is the sequence for 1 and the sequence for 2 and the sequence for 3, and then the ancestral sequence would have been the original, okay, that these 3 would have come from. And so now maybe you can get a little bit better picture on, which ones really are. Is it 1 or 2 that are more similar? Is it 2 and 3? Is it 1 and 3? DNA-wise, which ones have more similarities? Well, you've probably started to pick that out by now. If you haven't, take your guess. But you probably picked that first one, right, where 1 and 2 are more similar. And so this just kind of shows you, if you go through and you put little tick marks all over to show where differences sort out, we had in this first one, we would actually have 6 different little tick marks showing us where things became a little bit different, which you couldn't have figured out from the slides. And then the other 2 both had 7. So if we apply Occam's razor and that the simplest explanation is usually the correct one, then we go with number 1 because fewer events is simpler. right? And this is how people used to had to do things. This is how scientists used to had to try to categorize things. It was pretty difficult. They didn't always get it right. So big challenge. So now what happens? This is what happens. Now we put the sequence into BLAST and we let BLAST kind of run the algorithms and show us which sequences are more similar to others. And so other than the people that had to write the algorithms, it's much simpler for us now just to kind of go in and use the computer to let us sort it out. And then on our last slide here, we just have a few more terms. So the first term is monophyletic, okay? Mono being one. And in this case, all descendants of one common ancestor, okay? And I know it's hard to see, but there's actually a little grayed out area around this group right here, okay? And all of these ancestors or all of those organisms would have come from A right here. A would have been the common ancestor and all of this group. So this is a monophyletic group, okay? If you look over here, paraphyletic, okay? Para usually means next to or adjacent, okay? So some of the descendants of one common ancestor. So now here is your grayed out box. It's a little bit light, so I'm going to draw a little ring around it, okay? So there's our box, right? And it says some of the common descendants from one common ancestor. Well, A is the common ancestor again, okay? But we're only looking at some of the descendants. We're looking at all of them over here, but since we included B, but we're not looking at these because it wasn't highlighted, then we're only looking at some of the descendants from A. And so that's why it's called paraphyletic. And over here, polyphyletic means we're looking at the descendants of more than one common ancestor, okay? So if we look, stop, there we go. Sorry about the wiggle there. Here is the colored box on this side. So descendants of more than one common ancestor, because if you look, here's B, right? And D and E and H, I, J, K, all of these came from B, right? All of those guys did. But the colored box includes L and M, and they came from F, right? Which actually came from C, and they didn't come from B. So this is actually, we're looking at a group that comes from more than one common ancestor, again with the line, and so that's why we call it poly, right? More than one. So monophyletic, okay, descendants of one common ancestor, paraphyletic, some of them, and then some other ones next to it from the one common ancestor. And then the polyphyletic is the descendants of more than one common ancestor. So I hope that helps you, and here are your practice questions. And it's Friday, so I hope you have an awesome weekend.